Okay, let's talk about gene expression. If I just backtrack one quick second, we were talking about on Friday the endoplasmic reticulum and ribosomes, which the ribosomes are the actual protein factories of a cell associated with the rough endoplasmic reticulum, not so much the smooth endoplasmic reticulum. The RAR has the ribosomes, which is why it looks rough, whereas the smooth does not. And instead of making proteins, the smooth ER is responsible for making fats and storing calcium, releasing calcium, and so on and so forth. We talked about that the other day. Okay, I want to talk more about proteins and the formation of them, which is something that relates to gene expression. So this is very important, and it's a very simplified description of gene expression. I could go way more in detail on it, but I will not do so. Okay. So let's read through and I'll explain it. Although cells synthesize many chemicals to maintain homeostasis, much of the cellular machinery are devoted to synthesizing proteins in large numbers. We talked about proteins the other day. Remember, they are responsible for about just anything inside of our body. Remember that sentence? If something's happening in the body, good chance a protein is doing it. Okay. So these proteins determine the physical and chemical characteristics of cells, therefore the organism. Easy example would be melanin. That's found in our skin and our hair. That is a protein that gives us our skin pigment, our hair color. It even gives us the color of our irises and our eyes. Process of gene expression involves the synthesis of proteins from an original DNA template and involves two processes, transcription and translation. Okay. Simply put, if you think of it this way, it's super simple. Gene expression can be thought of as using DNA, making RNA, and then making proteins, okay? Making RNA from DNA is the act of transcription. Transcribing an RNA molecule from a DNA template. Taking that RNA and making proteins is what translation is. That's the simplified explanation of gene expression, and it's the most basic, easiest way to remember it. So transcription is genetic information is transferred from DNA to RNA. That takes place inside the nucleus. Specifically, the nucleolus is responsible for that. We make that RNA from that DNA template, and then we remove that RNA out of the nucleus, take it to a ribosome, wherever that ribosome is, not really important at this point, but then we attach it to a ribosome, and the information contained in RNA is translated into a sequence of amino acids. Remember, amino acids are like letters in an alphabet. You put them together in a specific way, you get a protein. Or in other words, this is a conversion from the nucleic acid language, because that's what DNA and RNA are, nucleic acids, to the protein language, which obviously is what proteins are. And like I said, this is the simplified, easiest way I can describe it. If you were to take, like, I took a class, cell and molecular biology. We spent like an entire unit talking about these steps. Very complicated, but very important. So for now, we'll just move on. Moving on to our next organelle, the Golgi complex, Golgi body, whatever you want to call it, consists of a stack of flattened membrane bound cisternae. And each Golgi stack or series of cisternae has two distinct sides in the inner region. I like to think of our Golgi like the FedEx of our cell, UPS, whatever you want to call it. It's going to receive packages and deliver them where they need to go. Okay. The packages I'm talking about are proteins that were made by the RAR and the ribosomes attached to them. Or it could also be a fat, okay? Except it would not be coming from the RAR, it would be coming from the SER. The bubbles that break free from the RAR carrying proteins or fats from the SER, called transport vesicles, will carry those freshly made proteins or fats to what's called the entry phase or cis phase, cis Golgi, okay? and that's the side that faces the RER and SER. Okay, that's like dropping off the package at the UPS store. The middle part of the Golgi, what we call the medial cisterna or medial Golgi, is where you might add a carbohydrate to it to form what's called a glycolipid or a glycoprotein. A lot of times proteins need kind of altered shape, and we talked about how important shape is for proteins. Any altering of the protein or fat is going to take place in the middle part, kind of like when UPS attaches a barcode, attaches a tracking number, all that stuff. And then the transface or exit face is where that altered protein, altered fat is going to exit the Golgi altogether to travel where it needs to go. And it's going to be exactly what we saw with the ER, little bubbles break free, little vesicles. Now, depending on what we make, if it's a phospholipid, maybe it becomes part of the plasma membrane. So we talked about phospholipids last time. If it's a hormone, it might be released from the cell altogether by a process called exocytosis. Maybe it's actually going to be a fully functioning organelle like what's called a lysosome, which we'll talk about on the next page. So overall, key function of the Golgi is to package different molecules together, can be used elsewhere in the cell or body. So like I said, kind of like the UPS or FedEx of our cells.
So is that like the shipping?
What's that? I'm sorry.
Is that like shipping?
Yeah, exactly. All right, lysosomes, organelles that are about 1/2 to 1 micron in diameter, so they're very small, surrounded by a single membrane, formed from the Golgi complex, like I just showed you. Inside of that single membrane are as many as 60 digestive and hydrolytic enzymes. Remember, hydrolysis is a chemical reaction that causes breaking down. Okay, so these I like to call the garbage disposals of the cell. They break down a wide variety of molecules once fused with lysosomes. So what they do is hydrolyze proteins, nucleic acids, complex carbohydrates, and fats, all those organic compounds we talked about the other day. They digest and dispose of worn out organelles, a process called autophagy, where auto means self, phage means to eat. So like when mitochondria, for example, get worn out, they become enclosed by a membrane formed by the ER, forming a vesicle called an autophagosome, which then fuses with a lysosome, and then those enzymes go to work, digest the broken down mitochondria or whatever organelle it is. Once digested, the lysosomes return the digested components to the cytosol for reuse. So we're recycling, not just completely pitching them. So that's one way old organelles are continually being replaced. We can degrade foreign material brought into the cell, destroy the entire cell, process called autolysis. And some lysosomal enzymes operate in what's called extracellular digestion, which is where the lysosome releases those enzymes out of the cell, which is a great way that, for example, our sperm help penetrate an egg to, make a baby, if we're talking about, sexual reproduction. Probably the most prominent time that we talk about lysosomes are when our white blood cells, called macrophages, digest things like bacteria and viruses and that kind of stuff. They engulf them and then destroy them. So that's kind of what the third bullet there is talking about. Peroxisomes and proteasomes. Through these on the next slide here. Next couple organelles. Peroxisomes look like lysosomes in structure, but are smaller and carry out different functions. They're also known as microbodies, but you'll rarely, if ever, hear me call them that. Inside of them are oxidases, which are enzymes that oxidize. We learned that means to remove hydrogen atoms from various organic substances. We find a large number of these in our liver cells. And what they do is generate and degrade hydrogen peroxide, H2O2, which is like the hydrogen peroxide that you might have in your medicine cabinet at home, which is potentially toxic to us. So they actually make it, but then just like neutralize it right away. It's an important process I won't really get into. Neutralize free radicals. We talked about free radicals fairly recently, said that they are like a tornado that cause destruction to things. And they can lead to things like cancer. They detoxify drugs and alcohol. So along with the smooth ER, they help keep our liver clean and our body clean as a result. Decompose fatty acids is the fourth function. Proteasomes, barrel-shaped structures, destroy unneeded, damaged, or faulty proteins by cutting the long proteins into smaller peptides, which just means when proteins get big and break down, we just snip them and kind of recycle them. Okay, the final organelle we'll talk about are the powerhouses of our cell, what we call mitochondria, which is the plural form of the word mitochondrion. Term or phrase powerhouse of the cell refers to the fact that they are responsible for aerobic respiration, which we talked about just the other day, means energy production. But what does the word aerobic specifically refer to? It requires oxygen. Very good. Okay. The more active a cell, like our muscle cells, the more energy they require. So they will have more mitochondria. Pretty straightforward there. Structurally, they're pretty cool. They have an outer membrane and an inner membrane, so they're double membraned. But what's really cool is that inner membrane, if you open it up, we'll see that it folds inward on itself, which increases its surface area. The inner foldings of that inner membrane are what we call cristae. And if you zoom in at them, you see that the cristae are loaded up with enzymes, which are the proteins involved with the making of the ATP. If you've ever learned about the electron transport chain, a really sophisticated thing that we'll talk about later on in the semester, these are the enzymes involved with that process. Okay. You ever learned about the Krebs cycle or citric acid cycle, which is also known as, that takes place inside of the cristae in the fluid called matrix. Okay. So that'll be something else that we talk about towards the very end of the semester. Other reactions, other things important, I should say, in the matrix, it's filled with DNA. It's the only other organelle aside from the nucleus that has its own DNA. It's called mitochondrial DNA. We inherit from our mother, and I won't get into why that is. There's also, as you can see, those little blue dots in there, mitochondrial ribosomes, and I've already talked about enzymes. All right, what we're going to do really for the majority of the rest of the way is talk about the plasma membrane, which is the boundary of a cell, like the walls of this room. Okay. Primary structures that form our plasma membrane are phospholipids. We talked about those the other day, which we learned are amphipathic, meaning that they contain both polar, which means charged, and nonpolar, which means uncharged regions. The heads we learned were polar, and that then allows them to have an attraction to other polar things like our water, which we've learned before has a big, or is a big percentage of our body inside our cells in the cytosol, as well as outside the cells in the extracellular fluid. Okay. The heads are described as being hydrophilic, which means that they like water, whereas the tails, which are nonpolar, are hydrophobic, which means that they are water-fearing. Okay. Now, when you take a group of phospholipids and you expose them to an aqueous solution, like both inside our cell and outside the cell, they arrange themselves in a bilayer, which means that there's actually two layers, where the tails from each of those two layers point together, and the heads point away from one another towards the water, both inside the cell and outside the cell. This arrangement, once again, is called a lipid bilayer. And this should not be brand new information to you, because we talked about this just last week. As you can see in our figure here at the bottom, there are many, many other things. There's other fats like cholesterol. There's glycolipids. There's also plenty of proteins. Okay, so proteins and carbohydrates are also located in the plasma membrane, and the ratio between these components is dependent upon the function of the cell. Now, do not think of the plasma membrane quite like the walls of this room, which are very rigid, made up of cinder blocks, bonded together, and pretty much immovable without, a sledgehammer or something much stronger than that. The plasma membrane and those arrangements of fats, proteins, and carbohydrates is very dynamic. Okay, it resembles a sea of lipids, as it says there, containing many types of proteins. And we describe the relationship and the way that those things kind of float around and move around as what we call the fluid mosaic model. We'll talk about the lipids and proteins that make up the plasma membrane. We just talked about the phospholipids, which make up about 75% of the lipids in the plasma membrane. There are two other lipids, or once again, lipids, remember, are fats. Glycolipids, which are about 5% of the lipids in the plasma membrane, found only in the layer facing extracellular space. So they're only found on kind of the outside of the cell. Help with adhesion among cells. They may mediate cell-to-cell recognition and communication and play a role in cellular growth and development. Then you've got about 20% of the lipids in the plasma membrane. We call that fat cholesterol, which we talked about the other day as a form of steroid. Strengthens the plasma membrane, but actually at the same time decreases its flexibility. All right, let's talk proteins. Two types of proteins are found in the plasma membrane and are classified into one of the two categories, either integral or peripheral, which are based on where they're at embedded in the plasma membrane, whether they're embedded in the plasma membrane, really I should say. Integrals are located really kind of throughout the interior of the plasma membrane. Many integral proteins are also listed as transmembrane, where the prefix trans means across or through, and they span through the entirety of the bilayer. Okay, peripheral proteins are pretty straightforward. They're found on the periphery or the outside, either facing the extracellular space or the cytosol, or the intracellular space. Okay, let's talk a little bit more about integrals. Many integral proteins are glycoproteins, proteins with a carbohydrate group attached to the end that protrudes into the extracellular fluid. Together, both glycoproteins and glycolipids, which are fats with a carbohydrate attached to them, create this sugary coat known as a glycocalyx that each cell has and is unique. It's like each cell has a special coat around it, okay, allowing a glycocalyx to act like a molecular signature, enabling cells to recognize one another. Okay, so it'd be like if we all had a different colored jacket on and we recognize each other like that colored jacket. Okay. Aside from that, it also helps cells adhere to one another and protects them from being digested by enzymes in the extracellular fluid. So that's kind of the way that we classify proteins based on their location. We can also classify proteins based on what they do and provide as far as functions go to the plasma membrane and therefore the whole cell. And that's what we'll discuss here on this page. And the first two are very similar, channels and carriers, and we'll talk more about those here in a bit because they play a role in bringing stuff into or moving stuff out of a cell. Okay, the difference really relates to what happens to the protein when something moves through it. Okay, a channel is like the doorway that you guys use to enter this room. Okay, and that when you came in and when you leave, the doorway, not the door itself, the frame around the door does not change. Okay, channels are the way that typically ions move into and out of a cell, like sodium, potassium, chloride, because they are not able just to float through a wall like a ghost. Okay, now we describe ion channels as selective, meaning that they only allow a single type of ion to pass through. So you have what are called sodium ion channels that only allow sodium in, potassium ion channels that only allow potassium in. Okay. Carriers or transporters kind of do the same thing. The difference is it's not showing you two different carriers, excuse me. It's the same one, just what it looks like differently after a few seconds, minutes, whatever it takes to get the substance across, probably not even seconds. Okay. The substance, in this case, maybe it's glucose, whatever, binds to the protein. Notice the pore is open to the outside, and then it flips and opens up to the inside. So the key here is that the protein must undergo a physical change in shape. Okay. Now, some channels have a door or gate that only allows them to open up that pore in certain situations. As far as the channel goes, it does not change its physical shape like a carrier does. The next ones are called receptors. Integral proteins serving as cellular recognition sites. So this is like how, for example, the hormone thyroid stimulating hormone that stimulates our thyroid only acts on cells in our thyroid gland. Okay. The thyroid cells in our thyroid gland have specific receptors for that hormone, and that prevents the hormone from just interacting with any cell of any other organ. Okay. Chemicals that bind to a receptor are called ligands, which is what you see over there in our picture. Enzymes, we talked about those the other day, integral peripheral proteins that catalyze or speed up chemical reactions, either on the inside or outside of the plasma membrane. Linkers, pretty much exactly what they sound like. They help link neighboring cells to one another or to protein filaments inside and outside the cell. And then I've already kind of talked about these glycoproteins, glycolipids, which act like that molecular signature. Identity markers, what they're listed as here, to prevent, for example, white blood cells from attacking our own body cells. Remember, white blood cells, I haven't really talked about them much yet, but they are the cells that kill bacteria and viruses and other foreign things. Okay, we don't want them attacking our own body cells, so they can recognize and hopefully avoid going against our own cells. Unfortunately, that's not always the case. All right, let's talk about the functions of the plasma membrane, which I've kind of talked about some of these already. Acts as a barrier separating the inside and outside the cell, so they keep what's inside the cell in, what's outside the cell out, unless we choose to bring something in or out. Okay, helps identify cells to other cells. That's what I was just talking about, like the immune cells. Participates in intercellular signaling. What we'll talk about here for the next several slides is the flow of substances into and out of cells, which is essential to the life of a cell. We talked about this today, and when I was explaining why cells have to be so small. Certain substances must move into the cell to support metabolic reactions, while other substances produced by the cell must be moved out of the cell. All right, let's talk about cellular transport and some new terms, permeable and impermeable. Okay, the term permeable means that a structure permits the passage of substances through it, while the term impermeable means that a structure does not permit the passage of substances through it. Think of our plasma membrane kind of like a strainer or a colander, whatever you call it, something that most people keep in their kitchen to use for straining out things like water when you make macaroni and cheese, which we made the other day in our house. Okay, if you've never made macaroni and cheese, it's a pretty easy process. You boil water, you put noodles in, cook the noodles until they are soft, and then you drain the water out. So you take your pot of boiling water and noodles and you drain it through the strainer. Okay, what happens when you dump it into that strainer? Water goes through the holes in the strainer and down your sink, unless you don't have it in the sink. What happens to the noodles? Stays in the strainer. Okay, so you can say the strainer is permeable to water. It's impermeable to the noodles. Our plasma membrane has this same, what we call selective permeability, meaning it only allows certain things to pass through it, because the permeability of the plasma membrane differs for different things. Okay, it is highly permeable, which means that things can easily pass through it to things that are nonpolar and uncharged, like O2 and CO2, which is why there's a big purple arrow for those, and steroids. Okay, here's the thing about steroids we learned the other day: they're fats. The primary structures that form the plasma membrane are fats, phospholipids. Fats and other fats generally can slide right through each other. Think of it that way. Okay. The lipid bilayer is impermeable, and that's why there's a U-turn here with our arrows on the right. Two ions, which are things like sodium, potassium chloride. That's because they have a charge, and the tails in the interior of the plasma membrane don't like things that have a charge. And then you're talking big things, large uncharged polar molecules like glucose, because they are too big just to kind of float right through. So what do we need to get those in? A carrier or a channel, which is what I was talking about here just a little bit ago. Okay, So protein channels and carriers allow for these molecules to pass through them in a very selective manner. There is a little slight permeability to small uncharged polar molecules like water and urea. So it's kind of like water can sneak through, but very, very, very small amounts. Remember, those tails that make up the bilayer don't like water. They're hydrophobic. So it's very, very minuscule amount of water that can just sneak through. So for water, it's going to use a specific type of protein channel. We'll get to that here after a bit. Not quite yet though. Now, when substances move across a cellular membrane or a plasma membrane, they move by transport processes that can either be classified as passive or active. Passive processes involve the movement of substances moving down their concentration or electrical gradient using only their own kinetic energy. Key phrase in that sentence there, down their concentration or electrical gradient. Now, we haven't talked about what that means, but we're getting ready to talk about concentration and electrical gradients here on the next page. Active processes involve the movement of substances against their concentration or electrical gradient. Key phrase in that sentence is against their concentration or electrical gradient. In active processes, cellular energy, usually in the form of ATP, is necessary to drag the substance, and I like the phrase there, and it's nicely quoted, uphill. There is another form of active process or active transport process that allows substances to enter or exit a cell. This is called vesicular transport because it involves small spherical membrane sacs called vesicles. We'll talk about that here towards the very end of the lecture. Okay. So we've mentioned here on this page twice the terms concentration gradients, electrical gradients. Let's learn what those things are. Now, I'll bounce back to that slide a couple different points here to kind of talk about different things that were listed there. But for now, let's just go through this slide. So the selective permeability of the plasma membrane allows a living cell to maintain different concentrations of certain substances on either side of the plasma membrane. What that means is by allowing only certain things through, either in or out, we're going to end up with different amounts of stuff inside the cell compared to outside the cell. Okay. The movement of a molecule that has no net charge or nonpolar is determined by its concentration gradient of that molecule across the membrane. Okay. So looking at the figure here on the bottom left, we can say letter A would be the extracellular space, so outside the cell. Letter B would be inside the cell, the cytosol. This line here with letter I represents the plasma membrane. Okay. Is there an equal number of whatever these bubbles are? Let's say it's oxygen here in the extracellular space compared to what's in the cytosol. Hopefully you guys can all see that there's more in letter A than letter B, right? Okay, go through and count them up. They're not equal, okay? When we move things passively, which is typically the way that we're going to do it, we're going to move them what we call down their concentration gradient. We're always trying to create what's called equilibrium. What does the term equilibrium refer to? Balance, or yes, you guys are doing great. Great job. Okay. But instead of thinking of it that way, think equilibrium as equal. Okay, where numbers are the same on both sides. Okay. So when you see the term or phrase down their concentration gradient, down the gradient, what that can be thought of as from high concentration to low concentration. Think of it like rolling a ball down a hill from an area high to an area of low. And as it said there in our lecture, only using their own kinetic energy, something that does not require ATP. Whereas against the gradient from high to low. Sorry, that's right. From low to high. And this requires ATP. Think of it like pushing a ball up the hill. Requires that energy to continuously get it against the force of gravity. So looking at this figure on the bottom left, let's say that we're going through passive transport, which means that we're going from high to low. So which direction would the molecules of oxygen go? From left to right, A to B, or from right to left, B to A? A to B, from left to right. We're going down from high to low. Okay. That's for things like oxygen, carbon dioxide, which have no net charge, or nonpolar. Okay. The plasma membrane also creates a difference in the distribution of positively and negatively charged ions between two sides of the membrane. Typically, the inner surface is more negatively charged and the outer surface is more positively charged, which can be measured with what we call electrodes. If you place electrodes, measure electricity charge inside a cell versus outside the cell, you'll find that the inside of the cell is actually negatively charged. And that's because there's a lot of negatively charged things like ions inside the cell. There's not as many positively charged ions inside the cell. That's because there's a lot more positively charged ions outside the cell. Okay. That difference in electrical charge between two regions, meaning inside and outside of the cell, constitutes what we call an electrical gradient. That can be termed what's called a membrane potential, which we'll talk about on the next page. Now, when an ion moves, which an ion is once again charged or polar, it's determined by its electrochemical gradient, the sum or combined effect of its concentration gradient and the electrical gradient across the membrane. So let's talk more about membrane potential. Membrane potential results from an excess of negative charge on one side of the plasma membrane and excessive positive charge on the other side. Cells, and the key here is at rest, normally have a membrane potential we call resting membrane potential, which is abbreviated V sub M, where V stands for voltage, M for membrane. If you place an electrode inside and outside of a cell, you can compare the ratio of negative to positive charge inside and outside. And what you'll find is that most cells have a resting membrane potential of negative 60 millivolts. So they are negatively charged on the inside. But that number is only at rest because when stimulated, ion channels can open and this number can change. Okay. And so let's look at three ions that we'll talk a lot about. Potassium, K+, sodium, Na+, chloride, Cl-. Okay, and what we show in the graph here at the bottom for each of those is their distribution normally at rest. So let's start with potassium. Cytosol is loaded with potassium ions. not very much potassium outside the cell. And so, like we said, we typically go towards equilibrium down their gradient. In other words, from high to low, which was why the arrow is showing from the cytosol to the extracellular fluid, okay, which is what this figure is showing you here. Lots of potassium would want to go out. Okay. Now, what would happen to this number if potassium leaves the cell and travels to the extracellular fluid? Remember, potassium has what charge? K plus. Okay, so each potassium ion is positively charged. Is this number going to stay the same? Is it going to become more negative or less negative? Anybody have a guess? It's going to change, so it's not going to stay the same. It's going to become more negative. And that's because we're losing positive charges from the interior of the cell. And remember, positive and negative are together, they cancel each other out. Okay, we're losing positives to cancel out the negatives, so we'll become more negative. Okay. What about sodium? Na plus wants to come into the cell from the extracellular fluid, and doing so brings in positive charges, which makes this number become less negative, or it could even bring enough sodium in that we become positive inside the cell, which is actually what happens when we talk here in a few weeks. So next week, I should say, when we get into action potentials. Chloride is like sodium in that it wants to come into the cell, but chloride is Cl minus. So when it comes into the cell, it brings its negative charges, which would make this number become more negative. Why do I bring this up? Because this is what we do. It's all about moving ions whenever we are doing an action potential. Electrical excitability is a property of a cell to rapidly change its membrane potential. In other words, we're going to talk a lot about action potentials as a result of a stimulus trigger. Whenever you touch something and it feels warm, ions are going to move, causing action potentials to travel in our body. Whenever you send a signal down to cause your muscles to contract, ions are moving. Whenever you have a thought, ions are moving. So whenever the nervous system is working, which is the first system we're going to talk about starting next week, it's all about ions. So you'll have a lot of fun discussing those, I'm sure. Okay, so now we kind of have a good idea of the things that dictate movement and then the ways that they take place, either active or passive. Let's talk about the specific types of movement across the membrane. First one is called diffusion, passive process in which the random mixing of particles in a solution occurs because of the particle's kinetic energy. Both the solutes, remember solutes are things dissolved in a solvent, which in our body, and whenever we talk solvents, typically water is the solvent, okay? So like the salt water that we talked about previously. So both solutes and solvent undergo diffusion. If a particular solute is present in high concentration in one area of a solution and low concentration in another area, solute molecules will diffuse toward the area of lower concentration, as we said, from high to low, which means they move down their concentration gradient. After a period of time, particles become evenly distributed through the solution, and the solution is said to be in equilibrium, or they're in balance, as we've talked about, equal numbers. Particles will continue to move about randomly due to their kinetic energy, but their concentrations will not change. Easy demonstration of fusion is taking a glass of water and dropping some colors, or dropping some food coloring in it. Okay, what do you see immediately? The food coloring starts to just move about the droplet or the glass of water. Eventually what you have is a glass of water that's partially pink colored. It might not be, depends on how many droplets of food coloring you put in there. Another easy example would be like spraying some Febreze. If I were to sit here and spray Febreze from the front of the room, okay, you guys in the back room wouldn't smell it, But Haley sitting right next to me would smell it immediately, be very strong, but over a few minutes, people in the back would start to smell it. And then eventually the smell would kind of fade away as it, spread out throughout the entire room. And, if I didn't spray that much, it would pretty much be gone within a few minutes. Okay. Several factors influence the diffusion rate of substances across the plasma membrane. The steepness of the concentration gradient. The greater the difference in concentration between the two sides of the membrane, the higher the rate of diffusion. So in other words, the bigger the difference between each side. Okay, if you've got a lot on one side and very little on the other side, you have a steep hill. Okay, ball's going to roll faster down a steeper hill than if they're almost already in balance. Okay, when charged particles are diffusing, the steepness of the electrochemical gradient determines the diffusion rate. Going back to my example of food coloring in water, what's cool is you can experiment with this, showing how temperature affects diffusion. Okay, take a glass of cold water and a glass of hot water, drop food coloring in each. Diffusion is going to happen a lot faster in the hot water than it does in cold. Good thing about our body is it's pretty warm, and so diffusion is really maximized because high temperatures, faster rate of diffusion. The mass affects diffusion. Okay, the bigger it is, the slower it moves, which is pretty much the case for a lot of things. Surface area, larger membrane surface area available for diffusion, the faster diffusion rate. We talked about microvilli the other day, the things that look like fingers projecting out of a cell, increases surface area, which maximizes diffusion. And then the distance with which diffusion has to take place, okay? The greater the distance over which diffusion must occur, the longer it takes. It's like, taking 50 steps versus taking 5 steps, okay? Obviously, taking 50 steps, it's going to take a lot longer than taking 5 steps. Places like our lungs where we have to do really rapid, really quick diffusion of oxygen into our blood, CO2 the opposite direction. It's so amazing how thin the walls of our alveoli are, the capillaries, which are the blood vessels that surround alveoli, which are the little bubbles inside our lungs, where we actually do diffusion. Okay. So diffusion across the plasma membrane takes only a fraction of a second because our membranes are so thin. Okay, there are several forms of diffusion. The first of which is the simplest, which is, I guess, probably why it's called simple diffusion. This is how, for example, nonpolar small molecules like oxygen and carbon dioxide move across a plasma membrane. Think of them like a ghost traveling through a wall. They do not require a transport protein. Neither a channel or a carrier is required. They can travel directly through the plasma membrane. Okay, and they are always going to do so towards equilibrium, which means from high to low. We can describe simple diffusion as direct unaided movement of molecules into and through the lipid bilayer in a direction dictated by the difference in the concentration of the molecule on the two sides of the membrane. Okay. Other molecules aside from oxygen and carbon dioxide capable of doing this are nitrogen gases, and then fats, fatty acids, steroids. Remember, fats can travel through fats, and fat soluble vitamins A, D, E, and K, which are basically fats as well. For most things, and like us, to get in and out of this room, we can't float through the walls. At least, I know I can't. We have to use an opening, like a channel or a carrier, which means we require help to travel in and out of this room. Another word for help is facilitate. So facilitated diffusion is diffusion with help. Transport proteins move molecules down their free energy gradient, either concentration, electrical, or both, in a process called facilitated diffusion, or it's also known as passive transport because it requires no energy. Two forms of facilitated diffusion exist. It's as simple as, if it's a channel doing it, it's called channel mediated. If it's a carrier doing it, it's called carrier mediated. Okay. Channel-mediated diffusion, therefore, involves small movement of a solute down its concentration gradient through a membrane channel. And this is how we typically are moving ions, because most channel-mediated facilitated diffusion involves ion channels, allowing passage of small inorganic ions to hydrophilic to penetrate the nonpolar interior of the lipid bilayer. Channel is said to be guided, which is like having a door covering the opening. Part of the channel protein acts as a plug or gate, changing shape in a way to open the pore and another way to close it. Facilitated diffusion through a channel is a very fast process. For example, more than 1 million potassium ions can flow through a potassium channel in 1 second. Carrier-mediated diffusion involves a carrier moving a substance down its concentration gradient. In this process, a solute binds to the carrier on one side of the membrane, released on the other side after a change has occurred to the carrier protein. And the rate at which this form of diffusion occurs is determined by the steepness of the concentration gradient across the membrane. Looking at the 3 figures here, or this figure, which got kind of 3 parts, the simple diffusion, like this, how oxygen can come into the cell from high to low directly through, is simple diffusion. Here's channel mediated using a channel. For example, maybe this is sodium coming into the cell from high to low. And for example, this is how glucose comes into our cells through a carrier or transporter, same thing. Once the channel, or excuse me, carrier transporter has physically changed its shape, going from high to low. There is another form of diffusion, and that's called osmosis. This is a form of diffusion specific to water. And here is where I will call it and record the rest, because I can't get through all this in 2 minutes. So I will start with osmosis of water in my next lecture, my next video, I should say. And have that posted shortly.